Hi, I'm Erica Ingram. I'm your host for the 1516 Biology Curriculum Overviews. Today we're focusing on Unit 5, Cellular Processes of Photosynthesis and Respiration. Now we have our instructional focus document. TRS has allotted 10 days for this unit. However, for GPIC, we have 8 days, starting October 26th, running until November 4th. In the black, we have our processing skills as noted, and our readiness and supporting standards are noted here. Red, of course, is the readiness, and green are our supporting. As you can see, there are a number of SEs that will be embedded throughout this unit. However, the main two SEs that I'm going to be focusing on throughout this curriculum overview is B.4B and B.9B. And now looking at our prior to the unit, as discussed in our previous overviews, we know that we use our prior to the unit to develop our engaged activities. So you'll be able to go through and read where the students are based off the information they should have knowledge of coming into biology, as well as the information that they have acquired, skills and knowledge that they have acquired being in your class up to now. So you'll be able to jog their prior knowledge from beginning of this school year up to now. During this unit, we're going to be using a lot of processing skills, planning, investigating, explaining. You want the students to be able to formulate, testify hypotheses, collect and analyze data, because they will have questions throughout the STAAR exam where they will have to use these processing skills. So you want to make sure that you are incorporating the use of those processing skills as you develop your explore activities. And after this unit, the students will use cellular processes, photosynthesis and cellular respiration for upcoming units, including evolution, viruses and bacteria, plants, and body systems. So we want to make sure that they are prepared and they get a good foundation over photosynthesis and cellular respiration so they can be successful in upcoming units. All right, so Unit 5 has one PA for the entire unit. It will be done on the evaluate day, which should be day 8 in the lesson cycle. And we will be utilizing the modified PA that was developed last school year by the biology team. And as you read through, you can see that there are different methods the students can deliver their understanding of the content through a graphic organizer, Venn diagram specifically, an illustration comparing the energy and matter flow, or completing the QR code scavenger hunt. And the QR code scavenger hunt is available on the secondary shifted resources drop box. And I do believe, if I'm not mistaken, in the QR code scavenger hunt, the students are required in one of those questions to illustrate or provide the equation for cellular respiration and photosynthesis and tell how they're related. So this is our modified PA that we will be using. Right, Unit 5 PA1 has a number of SEs that can be chunked in here, which are also presented in the previous sections of this PowerPoint presentation. However, we're focusing on B.9B and B.4B at this moment in the presentation. Compare the reactants and products of photosynthesis and cellular respiration in terms of energy and matter, which is a supporting standard. For our readiness standard, investigate and explain cellular processes, including energy conversions, transport molecules, and synthesis of new molecules. And so now we're taking this supporting standard and applying it in context of the cellular process we're speaking of in our readiness standard. So, of course, the cellular process is photosynthesis and respiration. And in additional slides, you'll be able to see how the STAAR has tested these SEs in terms of the readiness standard, as well as you'll be able to see how to deconstruct the SEs and better understand them so that you can teach the TEKS as it's written. Please make sure you adhere to the specificities that's provided for both of those SEs. Okay, real quickly, we're going to look at the data for our district on our Q2 summatives, and I'm going to focus specifically on B.4B and B.9B. However, 4A, 9A, and 9D are also embedded throughout the unit. So please take a look at those that data on your own. Specifically for B.4B on level has shown a little bit of growth over the past couple of years from 13-14 to 14-15, going from 35 to 41%. PAP was pretty stagnant. Looking at B.9B, 40% for on level. In 14-15 we have 44%. And pre-AP showed more growth on B.9B from 13-14 from 46 to 63%. So you want to use this data to guide your instruction and see exactly how we need to set our goals for this upcoming school year based off where we were in the past couple of years. Looking at STAAR data, we can see for B.4B and B.9B, we have gone like a little roller coaster. We go up, we go down, and then we go back up. Same thing again for B.9B, which it was not tested in 2012 as a supporting standard. But again, we started low, we increased in 14, and then we went back down in 15. So we want this curve to continuously to go up versus going in our current trend of up, down, up, down. On the science PLC observation form, we look for evidence of deconstructing the SEs. So right now I want to take the time to demonstrate what it's actually meant and how this process looks so that you can effectively use it in your PLCs. We're going to start with SE B.4B, and you can get a sheet of paper if you want to go along with me through this in deconstructing or breaking down this SE. So I'm going to read it first. Investigate and explain cellular processes, including homeostasis, which has been struck out, energy conversions, transport of molecules, and synthesis of new molecules. So when you deconstruct your SE, you first want to start with the verbs. Your verbs, this is what you want to circle, and I have already done this in advance. So you go ahead and you'll circle your verbs, which are investigate and explain. And then you want to look at your content. Well, we are investigating and explaining what? What is the content that we're focusing on? Well, we're focusing on cellular processes. And so throughout this biology unit, you'll understand and see that the cellular processes will be tested in different contexts. In our context that we'll be testing photosynthesis and respiration is in energy conversions, in transport of molecules, and synthesis of molecules. It will make a whole lot more sense when we look at the STAAR questions and we break them down to see exactly what they're testing and how they're testing it. And so then you want to look at what the verbs actually mean. I have provided the actual definitions of the verbs from Kilgo's training, and it is provided on the secondary shifted resources Dropbox. So we can look at our investigate definition: to observe or inquire in detail, examine systematically, and explain means to make plain or clear, render understandable or intelligible, to make known in detail. And so the words will actually be tested in terms of what these actually mean. So when you say to explain, to make it clear, to render an understandable or intelligible meaning, okay, to make it known in detail, explain it to me, tell me exactly what it is. And so let's look at the next SE. Okay, and now looking at B.9B, we can deconstruct, this SE and break it down to its parts. So compare, we're going to circle compare, which is our verb. Okay, compare what? What is the content that we're comparing? We are comparing the reactants and products of photosynthesis and cellular respiration. Okay, so that's our content, and I have put it in gold, but you will underline your content. And then in what context? Well, we're going to compare the reactants and products of photosynthesis and cellular respiration in terms of energy and matter. So now I have my verb, I have my content, and now I know how I'm going to compare or use this content in context. Okay? What does the verb compare mean? Well, compare means to examine two or more objects, ideas, etc., in order to note similarities and differences to consider or describe as similar, equal, or analogous, liken. And again, this definition comes from Margaret Kilgo's strategies. Okay, and so we're going to look at the 2015 question 22 from the STAAR exam this year. And I got these questions again from Leap Forward, which is the 2015-16 biology release test. And so now you can see as we go through how the state is testing the SEs exactly as they are written. So it says cells can generate as many as 36 to 38 molecules of adenosine triphosphate, ATP, from the metabolism of one molecule of glucose, which cellular process results in this amount of ATP production. And so we know that the process, cellular process where glucose is converted to ATP is respiration. So we're looking at respiration. Okay, and we also know that it's testing the cellular process respiration, but in what context? Is it energy conversion, transport of molecules, or is the synthesis of new molecules? How are they testing this concept right now? Well, they're testing the concept in terms of two things, I believe, which is energy conversion as well as the synthesis of new molecules. Well, Erica, why do you say that? Glad you asked. Well, it says here that you generate as many as 36 to 38 molecules of ATP. Okay, you're generating, you're producing, you're making a new molecule, and this molecule is coming from glucose. So you're going from a molecule of glucose, which is the reactant, to produce ATP, which is the chemical energy in respiration. And so that is energy conversion as well as a new molecule being produced. And in Kilgo training, Margaret Kilgo instructed us not to focus on the distractors, but to look at the correct answer. And so the correct answer here is aerobic cellular respiration. And why do we know that it's aerobic cellular respiration? Because ATP molecules are being produced, and with the number that is being produced, we know that that requires oxygen. And so if oxygen is present, that means it's aerobic cellular respiration. Well, if there are a number of ATP molecules that are being produced, but they are of a low number, then of course that would be anaerobic cellular respiration. So this is how the state is actually testing the concepts in context. Yes, it just says in cellular processes, but the cellular process, the supporting SE, which is comparing and contrasting photosynthesis and cellular respiration, is a cellular process, and they're testing it in terms of these different contexts. So I hope that makes sense. And you can go through additional ones. I'll do one more with you, and then we can go through other ones real quickly where you can understand how they're being tested. Okay, next question from 2014 over the same standard. The question is, which group of organelles is directly responsible for the production of new molecules within a cell? So if you look back at the SE, tell me, you can't tell me directly, of course. So think about which type of process is being measured at this point. And I didn't mean to take out the including just the homeostasis. Energy conversion, transport of molecules, or synthesis of new molecules, or more than one. And based off the context, production of new molecules within a cell, it's clearly testing on the synthesis of new molecules. Well, what's the cellular process? The cellular process is cell structure and function. So now we're looking at the organelles, the different cell structures and functions in context of synthesis of new molecules. And so the correct answer is A, looking at ribosomes, endoplasmic reticulum, and the Golgi apparatus. So I hope you're starting to see how the state is using the SEs, developing the question, and they're testing the different parts of the SE in different contexts of cellular processes. Okay, on 2013 question 6, we're looking at the same SE. Question 6 is, which of these statements best explains the process of energy conversion that takes place in the mitochondria? So our cellular process is cell structure and function because we're focusing on the mitochondria. And how does the mitochondria work with energy conversion? So they're asking us about this cell structure in terms of energy conversions. And we look at our correct answer, which is J. And as we can see across the state, 67% of the students understand the function of the mitochondria. We still have some discrepancies that are shown from F, G, and H. So these are our distractors. We don't want to look at those, but look at how they're asking the students and look what answer they're looking for. The energy in the bonds of glucose molecules is transferred to the phosphate bonds in ATP. So yes, in respiration, that's where it takes place in the mitochondria. Glucose goes from being broken down into ATP, which is adenosine triphosphate. So it's very important that the students understand phosphate bonds are being developed in the process of going from glucose to ATP. Now 9B is the same process of compare the reactants and products of photosynthesis and cellular respiration in terms of energy and matter. So the question is, diatoms are one of the most common types of phytoplankton in marine habitats, like plants. So we know that they're photosynthetic if they're like plants. Diatoms contain chlorophyll and produce glucose from which of the following? So we need to understand where is glucose being produced. And that takes us back to understanding, okay, I need to go backwards and forward. I need to understand my products of photosynthesis and my reactants of photosynthesis. So if glucose is my product, how do I get glucose from a plant? What does a plant need in order to produce its food, which is glucose? It needs sunlight, it needs water, and it needs carbon dioxide. And so I want to look and see which of those, which is my correct answer, carbon dioxide and H2O. And so you can see now that the students will have different contexts and different situations. Yes, you may not can go through all the different types of examples, but it's the concept that they need to be able to apply to different situations. And if they understand the concept, then they can answer the questions in different contexts. Okay, in 2014, they assess using a graph, and every time students see a graph on an assessment, they just, for some reason, freak out. They make an assumption that they don't know how to analyze it, or they'll go into just guesstimating, as I like to say. But we can see the 54% of the students across the state got this question right. The correct answer was F. And I'm going to read the question real quickly. Most plant leaves take in more carbon dioxide as light increases. They give off carbon dioxide if light intensity is too low. The graph shows the plant's carbon dioxide levels at different light intensities. And so they're breaking off this graph to show you from this gray portion, this is where carbon dioxide is taken in a plant. And this is the section, smaller section below, where carbon dioxide is given off. So it's just a point of thinking about, hey, plants take in carbon dioxide when? Well, they take in carbon dioxide in photosynthesis because they need it to produce their food. So this is my way of writing photosynthesis. Y'all bear with me. And when do plants give out carbon dioxide? Well, they do that in cellular respiration. And so now the question is, at point X, the rate of which process is equal to the rate of photosynthesis is here. So if this is carbon dioxide being taken in, this must be photosynthesis. If this is carbon dioxide being given off, this is cellular respiration. So what is this point where they intersect if this point is X? So the point must be intersecting at cellular respiration and photosynthesis. And F is our answer. So they're using a processing skill. Yes, it takes more thinking, but this is how they are testing our students. This is how they are getting them to analyze graphs and to break down exactly what the question is asking them and not what they are assuming a question is asking them, or how different ways carbon dioxide taken in or light can affect the plant. It's not asking anything about that, which are distractors that they use to throw the students off. And as you can see, in some ways that they were successful. So let's try to make sure our students are not thrown off and use deductive reasoning based off of what is actually being asked in the question. In 2013, how did the state test question 12? And so, which of the following correctly describes how a diagram of cellular respiration would differ from a diagram of photosynthesis? So now we're comparing and contrasting diagrams. Okay, so they did not give me a diagram. I should have a visual of a diagram in my head based off of what I know about the reactants and products of photosynthesis. Okay, and so automatically I know that I should see sunlight for photosynthesis. It's my solar energy or radiant energy or light energy, in whatever terms you want to use. I should have carbon dioxide. Okay, I should have some water. Okay, and I am going to produce glucose, and I'm abbreviating GLU, and oxygen, because plants give off oxygen. Okay, so this is my sun, this is my carbon dioxide, and this is my water. Okay, producing glucose and oxygen. Don't judge me. Thank you. And so that's photosynthesis. And cellular respiration, I'm going to take this glucose and oxygen, which are my products, and make them my reactants. So I have glucose plus oxygen, and this is going to produce ATP plus exhale, give off CO2. So I go from light energy to chemical energy. And what's my main source of getting this energy? Glucose. So when I look at my answer choices, I see that cellular respiration diagram will show that glucose is the main source of energy in cellular respiration, which is correct. Main source of energy. Sugar molecule converted to energy. And looking at our testing, the correct answer was G, and 48% of the students across the state got that correctly. And you can see the second highest was H. So there's a huge misconception here as to why a cellular respiration diagram, which the mitochondria is responsible for cellular respiration, and the chloroplast for photosynthesis, why would all of this be taking place inside of a large protein? So there's a misconception there that we want to make sure is addressed throughout our lessons. And once again, your essential questions, we definitely want to make sure we're using those to drive our instruction. Your essential questions are the precursors of the students understanding the PA and being able to effectively and efficiently do the PA with fidelity. if the students can answer these questions, they can do or answer whatever the PA is requesting of them. So you want to make sure you're utilizing your essential questions. You can use them in your explain portion. You can use them in the elaborate. Your in-lesson guiding questions, of course, you know these are your guiding questions to probe the students, and you can use these along with others that you come up with on your own, but you want to make sure you're embedding those throughout your lesson cycle and questioning the students and not giving them the answers. Thank you. Okay, in this unit, we do have some repeating vocabulary terms from previous unit. Chlorophyll, of course, chloroplast from cell structure and function, but the majority of the terms that are being used are new to the students. So cellular respiration is not new, photosynthesis is not new, but ADP, ADT cycling, aerobic respiration, anaerobic, ATP, Calvin cycle, chemosynthesis, and additional related vocabulary terms you can find in your IFD. Those are all new terms that the students will be introduced to in this unit. You have a list of misconceptions that you want to definitely make sure you are addressing through your explore activities and give the students to explain their understandings in the explain portion. And there you can see if any of these misconceptions still lie in addition to any others, and you can clarify those after the students have explained their understanding. Now we are at the end and we are ready to go through our checklist and make sure that we have everything necessary to effectively plan a 5E lesson cycle and teach the TEKS as they are written. So we've studied the Unit 5 IFD, looked at our performance assessment, looked at our overview. We've looked at the district and STAAR data from previous years. We've deconstructed the SEs and then taken those deconstructed SEs and analyzed STAAR questions to understand how the SEs are being assessed. We've looked at our essential questions that we know we're going to use throughout our 5E lesson cycle, what the important vocabulary terms are that needs to be incorporated throughout the lessons, and develop and explore activities to address any misconceptions. So now we're ready to plan our 5E lesson cycle. Remember, we have 8 days for Unit 5, and just as a reminder, make sure that you are planning each E over the allotted days, not each E being done every day. If you have any questions or any concerns or anything that you may need, please do not hesitate to contact me or Michael Brinkman. Thank you, and I hope that this Unit 5 curriculum overview has been helpful. Have a great day.